Hello, everyone. Welcome to today's live broadcast, Real-Time Profiling of Tumor Heterogeneity in Metastatic Disease. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Epic Sciences. Epic Sciences develops new, highly sensitive diagnostic tests that identify and molecularly characterize circulating tumor cells, CTCs, in the blood to provide a better way to detect cancer and personalize and monitor treatments. For more information, visit epicsciences.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the Q&A button, the green one, green Q&A button, lower left. I would now like to introduce today's speaker, Mark Landers. Mark Landers has 18 years of experience developing molecular assays with extensive experience in qPCR, microarray, and NGS assay development in both research and regulated environments. He has led the commercialization of several microarray, qPCR, and NGS-based assays for biomarker discovery. In his current position as Director of Genomics at Epic Sciences, he is leading the development of assays to characterize CTCs using genomic endpoints, including QRT, PCR, and NGS. Prior to his tenure at Epic, Mark was head of R&D at AltheaDx, where he developed several companion assays for targeted therapies. He has also held several senior-level scientist positions in the San Diego biotech community at companies including Life Technologies, Serogene, and Vical. I will now turn it over to Mark Landers for his presentation.
Thanks, Judy, and thanks to all the participants today for joining us. The focus of this talk is tumor heterogeneity in metastatic patients. So during this webinar, we'll first review the concept of tumor heterogeneity and the use of circulating tumor cells to profile heterogeneity in metastatic disease. We'll give you some background on the Epic technology platform and its key differentiating factors to detect and characterize these heterogeneous CTC populations. And most importantly, we'll show you some of the heterogeneity we see in late-stage metastatic patients, and we'll use data from a cohort of metastatic castrate-resistant prostate cancer patients. So before I do that, let me give you a little background on Epic Sciences. Epic Sciences, our mission is to improve patient lives by enabling precision medicine with non-invasive real-time tests that can profile rare cells in cancer. Simply put, using our proprietary CTC platform to better characterize a patient's disease, characterize the subclonal diversity present prior to and throughout the course of therapy, allowing us to tailor more accurate and efficacious therapies. So Epic was founded in 2008. It's a San Diego-based company. We're up to 117 employees now. Shown in the bottom left is a picture of our 40,000 square foot facility in San Diego. We have a fully licensed CLIA lab that we can use to run these assays in support of clinical trials. And we're in the process of developing IVD instrumentation for diagnostic purposes. So currently we offer the Epic technology as a service in our lab, and we currently have over 26 partners, pharma partners, and 16 academic partners. We have contracted to analyze over 15,000 samples and have participated in 45 clinical trials across 12 different cancer types. We have developed 45 different protein immunofluorescent assays targeting different molecular targets. We've developed a lot of DNA fish assays targeting DNA rearrangements that can be targeted in individual CTCs. We're developing our RNA fish capabilities, And most recently, we've developed NGS capabilities to do next-gen sequencing on single cells to profile genome-wide copy number variation. So this is a diagram of the drug development cycle and how EPIC can use its platform to work with our pharma partners throughout the entire process. So to develop assays, early on we can help our pharma partners to develop assays for observational trials where we are looking to better characterize pharmacodynamics, mechanism of action, mechanism of resistance, and identify markers that could potentially be used for CDX applications, companion diagnostic applications. We can take those assays and qualify them and validate them and run them in our CLIA lab prospectively for patient selection to support interventional clinical trials. And then moving forward, we're developing the capabilities for diagnostic applications where we could use this for both companion and novel diagnostic therapy selection and monitoring of drug resistance. So we really think one of the most exciting applications of this technology is real-time monitoring of disease resistance or drug resistance and seeing those emergence of resistant clonal populations. So one of the biggest problems facing precision medicine today is tumor heterogeneity and its negative effect on therapeutic efficacy. And so cancer is a heterogeneous disease. There's been a lot made of that. Heterogeneous both in patient-to-patient comparisons. Within a single cohort, we have a lot of different drivers in tumors of the same tumor type. As well as within a single tumor, we have lots of subclonal populations of cells that potentially have different genomic drivers or different phenotypic features that make it very difficult for drug development and patient stratification when you're starting from traditional tumor biopsies. This was a paper published recently. This was Charlie Swanton's group where they looked at, they wanted to further investigate intra-tumor heterogeneity and the subclonal diversity within tumor samples. So they went in and mined TCGA data from 2,700 approximately single tumor samples across 9 major cancer types. And they took the variants that were called, the single nucleotide variants, as well as the copy number alterations that were called, and based on their allelic frequency or the fraction of the mutant allele in the sample, they could define it as clonal or subclonal. And then they could further characterize the relationship of those mutations as being early or late and designate them as either clonal or subclonal drivers of disease. And so some of the important findings of this study were that there were frequent observations of subclonal drivers present in every tumor type they looked at. So subclonal populations that have different drivers within a single patient. If you look at this figure shown here on the right, each column represents a different tumor type that they looked at. Shown in each row are therapeutically relevant gene targets that are often used to select therapies or stratify patients. What you can see here are, highlighted in blue are the samples, the genes in which there were significant high frequency of subclonal populations in those samples. So was there a lot of heterogeneity in the targetable mutations within these different tumor types? And the answer was yes. And one of the main conclusions that they drew from this study was the need to stratify targeted therapy response according to the proportion of tumor cells which have the driver. And moreover, to go even further and let the proportion of tumor cells that have an alteration within a particular pathway that might be druggable. So there's been a lot of other reports showing that intratumoral heterogeneity can have a negative effect on the efficacy of targeted therapies. This was a publication from 2013. This was whole exome sequencing of 149 CLL patient samples. They did both looked at single nucleotide variants as well as copy number variants, And based on the relative abundance of the mutant allele in the sample, they were able to categorize these as either clonal or subclonal. And then they were able to compare that across the patient populations and in respect to response to the therapeutic as well. And so the interesting take-home from this message, in the upper left-hand corner, you can see the average number of mutations per sample that were detected, either subclonal in blue or clonal in orange. So there were more subclonal alterations detected in patients that had received previous treatment versus the patients that were treatment naive. And that was a statistically significant difference. And so if you take those patient populations and you stratify them by, yes, there was subclonal populations present, and that's shown in the dark green line on this Kaplan-Meier curve, or there were subclonal drivers, there were no subclonal drivers detected, the light green line on the Kaplan-Meier curve, and you make two separate distinct populations and stratify based on just subclonal drivers, you can see that the time to therapy failure was much shorter for those with subclonal alterations present. So this, in summary, you could just think that the more heterogeneity you observe, the worse the therapeutic response. So another interesting study is we know that there's tumor heterogeneity. There was a paper published in 2013, and this was a study to look if the CTCs could reflect the subclonal diversity that we see in the tumor population. And so this is a pilot study they did, and they used EpCAM-enriched CTCs as well as primary tumor biopsies and metastatic tumor biopsies, and they did whole exome sequencing. And so here what they wanted to see is, could they identify subclonal alterations in the tumor samples that were reflected in the CTCs. And so the alteration shown here on the right, and the top is the primary tumor, and in the middle is the metastatic tumor, and then the individual CTCs that were sequenced are shown below. And the mutations that are highlighted in red, those are the alterations that are common to all three. They were in the primary, they were in the metastatic, and they were in the CTCs, and these can be considered early driver alterations. Whereas if you look in the blue, those were specific to just the metastatic tumor biopsies as well as in the CTCs. And then if you look at the black mutations that were highlighted there, those are specific to CTCs. And so they didn't find those originally in the metastatic or primary tumor biopsy. However, they went back and they resampled and they resequenced, and they were able to find low subclonal populations of cells with that mutation in either the metastatic or primary tumors. So, you know, it really speaks to the fact that you can use CTCs to monitor subclonal heterogeneity at even very low levels, and they do reflect the active metastatic population. So it also highlighted the power of single-cell sequencing and being able to look at single CTCs. And this, I couldn't resist putting this in here. This is a slide that Levi Garraway presented at AACR this year, and it's really just an analogy of comparing the analysis of single CTCs versus doing more bulk approaches of either pooled CTC population, cell-free DNA, or starting from, say, a tumor biopsy. So if we really want to understand the diversity of clonal populations, if you look on the left, I can characterize each one of these, and I can get a much better idea to their relative proportions, what their characteristics are, and I can monitor those subclonal populations. If I mix them all up, and then try to decipher which populations are there, it makes it much harder. So at this point, I think I'm going to switch back to reviewing the EPIC technology platform and why we think it has some key differentiators that allow it to differentiate it from other technology platforms for the analysis of CTCs and allow us to look at more heterogeneity. And so how does it work? How does the EPIC analysis work? So starting from patient samples. So this is showing you our EPIC lab in San Diego. and our LabCorp partners in either Belgium or Singapore. So blood is taken at the clinical site, and it can be shipped to any of these 3 destinations. For North American and South American trials, blood is taken in a 10 ml tube, shipped to our San Diego location at ambient room temperature shipping conditions, and then is processed. For LabCorp in Belgium and Singapore, if you're in Europe, you can do the same process. However, they will just do the first stage of the process to get the samples to a stable stopping point, and I'll show you more about that in a second. So this is kind of a diagram of what the process is. And so blood is received at the laboratory, and it's processed. So the 10 ml tube of blood is processed where we lyse all the red blood cells, and then we pellet out all the nucleated cells and resuspend them, and then plate them onto replicate slides. And so these are proprietary slides that help us get an even monolayer of about 3 million cells per slide. From a single tube, we can make 12 slides, and then these slides are stable at this point and can be stored at -80. And so this is where our LabCorp partners stop, and they can now ship us those samples, and they're very stable for shipment and for subsequent analysis. So now we can go into our -80 freezer, and we can pull out our two slides per assay is typically what we use, but you could do more or less. We pull out 2 slides, and then we want to identify the CTCs. So we use a cocktail of immunofluorescent antibody staining targeting cytokeratins, CD45, and DAPI. So CD45 is specific to the blood cells, cytokeratins are specific to epithelial cells, and DAPI tells us which cells have intact nuclei. We also have an open channel which we can mix in a 4th marker. So an example of this, I'll show you in greater detail some examples, different examples, but just so you have a good reference point, we can mix in, if we're working in prostate cancer, we would typically target the androgen receptor, or if we were working in breast cancer, we could add HER2 or ER. So after we do the staining for these different molecular markers, we can then scan the slides and take the images. And so after scanning, the images are processed, and we don't only look at the molecular markers. We have a proprietary algorithm that looks at cells pretty much the way a pathologist does. It was trained by pathologists. And so we look at a lot of cell morphological features that help drive the identification of cells that are different in a field of 3 million white blood cells. So by measuring these morphological features and integrating that with the molecular markers, we can then come up with candidate CTCs that are then confirmed by trained operators here. And those are generated into a report. So why is this strategy so different and enable us to see so much more clonal diversity than other platforms? And so the majority of other platforms on the market rely on some sort of physical enrichment strategy to separate the circulating tumor cells from the contaminating white blood cells. And so, most of these are relied on a EpCAM enrichment or use CK to enrich, but basically antibodies targeting cell surface markers that are specific to circulating tumor cells, or they filter by size, which makes the assumption that circulating tumor cells are always going to be bigger than white blood cells. And so all these approaches have the same similar issues where you're going to have CTCs that don't necessarily have epithelial markers presented on the surface, or you're going to have CTCs that can look at that look about the size of white blood cells and would blow right through a filtration device. And so the key differentiating factor of the EPIC platform is that we look at all nucleated cells within the sample. And so there's been a lot of reports recently about EpCAM positive, EpCAM negative cells, circulating tumor cells, and when we see these cells, we can tell that they're also expressing a biomarker of interest. And I'll go into a little bit more of what these look like, but I think this is what allows us to see the increased clonal diversity present in metastatic patients. So this is what it looks like. This is an EPIC slide, and we can zoom in, and you can see what a field of cells would look like. And so you see all of our white blood cells around that can be used as controls, and then you can see our rare cell right there in the middle. And so for each test, we analyze about 6 million cells to identify the rare cells in the population, and we use a multitude of morphological and molecular parameters as we previously described. But the key things to take home at this point are no enrichment, so we're not manipulating the cells, we're not losing any epithelial plastic cells, and we're not losing any smaller CTCs. And so we see a larger range of CTC subtypes. So we wanted to show you kind of some images of what those might look like and why they're important. And so if you look here, this is showing you what our normal controls look like. This is all the white blood cells that we see within the patient population. They have DAPI nucleus, and they're positive for CD45. The rare cells that we find, some are traditional CTCs. So these are traditional CTCs with epithelial markers that are expressing CK. They have an intact nucleus, and these are CTCs that would probably be detected by a lot of other platforms. However, we see a larger diversity than that. We see a lot of small CTCs, so these are CTCs that have epithelial markers sometimes. They have cancer genomes. However, they're just much smaller. They're approaching the size of white blood cells. We also have the ability to detect apoptotic CTCs, where we see that the nucleus is no longer intact, but we can also count these as a pharmacodynamic endpoint. So here's an example of a CTC that is in prostate cancer, and this is a CTC that is no longer, it's become epithelial plastic. It's no longer expressing cytokeratins, but it's still expressing its androgen receptor very brightly. And when we sequence these genomes, we can tell that these are actually cancer genomes, and we can really see that these would be cells that would completely be missed by most enrichment-based methods. So there are a lot of other morphological features that help us further characterize the CTC. So we can look at very detailed morphologies. We can see the speckling of cytokeratin. We can look for macronucleoli, which could be associated with nuclear instability. We could also look for clusters. And so this is a big advantage. We can detect clusters not only of traditional CTCs, we can detect clusters, small cell clusters, CK- clusters, clusters with macronucleoli, and we can further characterize these. And there's been a lot of recent publications showing that CTC clusters have been associated with an increase in metastatic potential and associated with poor prognosis. So there's a wide range of integrated analysis beyond just CTC enumeration. We have a variety of protein markers that we can use to further characterize these cells that could be potential biomarkers. And so we'll go through the list, or an example list of different protein markers that can be used. But the key point here is that all these endpoints can be used together in an integrated analysis. So not only are we looking at the cell morphology, what markers it has on the surface, we can do further detailed analysis looking at gene amplifications by FISH, where we're looking at DNA arrangements. This is your ALK. We can look at PTEN loss. We're starting to get online our RNA FISH to be able to look at the expression of specific transcripts in our CTCs by RNA FISH. And more recently, we've started developing our genomics capabilities to where we can go in and isolate particular CTCs of interest, recover them, and analyze their genomes for copy number alterations genome-wide, as well as targeted mutation panels. And so integrating all these endpoints together gives us further resolution to be able to define distinct subclonal populations. And here's some examples of some of the assays. This is certainly not a completely comprehensive list, but just to give you an idea of some of the protein assays we've developed, genomic assays we've developed, and what we're working on. We have protein assays that target some very interesting targets in prostate cancer. We have AR-targeted protein assays that target both ends of the AR protein. We also have targets towards PSMA. PSA, we have PD-L1, which is a particularly important marker in immunotherapy. We can target incoherent expression on the cell surface, which is a common EMT marker. Or we can look for genomic rearrangements, which are associated with tumor suppressor loss or oncogenesis, like PTEN loss, HER2, ALK. Most recently, we launched a whole genome sequencing copy number variation assay, and this is doing low-pass whole genome sequencing to look genome-wide for tumor suppressor loss or oncogene gain. And so this allows us to identify subclonal populations by genotype, and then we can integrate that information with our protein analysis and phenotypic analysis. We're working on launching some assays that target the ARV7, so this is a transcript, and I'll show you some data on this later in the talk. We also have new FISH assays that can target Aurora K, RB. We also have, we're working on developing targeted resequencing panels, so looking at therapeutically relevant point mutations within the genomes of our CTCs, so taking single cells and monitoring actionable mutations in the subclonal populations. And we're also developing qRT-PCR targeted gene expression, so looking at small gene panels starting from single cells, and we're rapidly developing that. So at this point, I just want to remind you that the key advantages to the EPIC platform are, enrichment-free CTC analysis, as well as integrated phenotypic, molecular, and genomic profiling. And these allow us to paint a really distinct picture of the clonal diversity that's present prior to and throughout the patient's treatment. So we also have the ability to biobank samples, which is for subsequent analysis, which becomes even more important because as you bank these samples and new biomarkers become interesting, you have the ability to go back to these samples later for subsequent analysis. So this is where we're going to dive into the data, and so I think we're going to start by looking at ARV7 expression in metastatic castrate-resistant prostate cancer patient samples. But before we do that, I just want to show some data that was presented last year from the Johns Hopkins group where they looked at ARV7 as a predictive marker for resistance to AR-targeted therapy in CTCs. So this was data they generated by EpCAM enrichment of CTCs from whole blood in a pooled CTC sample. They then lysed those open and looked for the ARV7 transcript by qRT-PCR. So the presence of the V7 transcript shown in gold, the patients that are positive for that, can be associated with poor response to the therapeutic or resistance to AR-targeted therapies. And so shown on the left is a waterfall plot, and the way to read these is that when the lines go up, that's the PSA level going up after treatment, and so these patients continue to resist therapy if their PSA goes up. If the line goes down, then you can see that the PSA is decreasing. The therapy was somewhat effective and worked to decrease their PSA. So these samples were taken at baseline, they looked for the ARV7 transcript, and then they looked to see if those patients then responded or didn't respond. And you can see that the assay is very specific to the patient's ARV7 was mostly detected in patients who resisted either enzalutamide on the left or abiraterone on the right, AR-targeted therapy. And this makes sense because the ARV7 doesn't have the doesn't contain the domain that either of these drugs target. So we wanted to look at a single cell assay taking an ARV7 antibody and then further characterizing the CTCs we see for the expression of ARV7 actually in the nucleus of our EPIC CTCs. So this is work we did in conjunction with Memorial Sloan Kettering and Howard Shear's group, and this is we took 60 baseline blood samples from patients prior to their treatment with either AR-targeted therapies or chemotherapy with taxanes. we took baseline samples prior to first line, second line, or third line treatments, and then the patient response was measured by their PSA pattern. Did the PSA just go up following treatment, or did they show some initial response to PSA? And so before we get to the association with the ARV7 expression and response, I think it's important to point out the wide variety, a biological range of CTCs that we see ARV7 expression in. So we can see that ARV7 protein is expressed in CK-positive CTCs, CK-minus CTCs, and CTC clusters. So this is a lot of these cells would be missed if we did EpCAM enrichment and looked at just for ARV7 expression. So we really feel like the single cell resolution and the larger range of CTCs we can see can paint a better picture of the true clonality of cells expressing ARV7 in resistant patients. And this is data showing the frequency of ARV7 CTCs within patients. And so shown here in the beehive plots on top are the relative number of the CTCs detected that were expressing ARV7 in the nucleus in patients that were sensitive to AR-targeted therapy and the patients that were resistant to AR-targeted therapy. Shown on the right is the similar curve for patients that were either sensitive to taxanes or resistant to taxanes. And the key take-home message here is that we only detected ARV7 protein in the nucleus in CTCs from patients that were resistant to AR-targeted therapies. We didn't detect any in the CTCs from patients that were sensitive to AR-targeted therapies. However, when we looked at taxanes, we detected in both resistant and sensitive patients. But that doesn't really paint the picture of true clonal heterogeneity. What we want to see here is if we can look at the relative proportion of cells that have that are expressing the V7 in the nucleus versus those that are not expressing V7. And so that's what the bar chart on the right is showing. Patients, each bar represents a patient. Patients, we're looking at ARV7 CTCs, the number that were quantitated, and so we're looking at first-line patients, then second line, then third line, moving from left to right. And so what should jump out at you is that the large heterogeneity of ARV7 expression in the CTCs. And if you go further and you look at just the percentage of ARV7 CTCs that are shown in the table below, you can see that in second line and third line patients, the percent of CTCs expressing ARV7 can range anywhere from 17 up to 79% of the CTCs. So clearly there is ARV7 being expressed in the CTCs, but however there's subclonal populations. And so it's important to have that kind of resolution to be able to figure out what's really driving the disease. And so as I mentioned before, we look at a lot of phenotypic features in our CTCs and to help us further characterize the subclonal populations. So we have phenotypic measurements that we take, and I think we went through that. But this is the process for better characterizing the CTC phenotypes. So we have, after the slides have been scanned, we can further analyze the image, and this is the digital pathology algorithm that we've developed that will segment the cells and start collecting a lot of data about the cell size, the cell shape, nuclear artifacts, that it measures a lot of ratios, and I'll go through these all in a little more detail. But the point is that we can really get a detailed picture of what the cells look like. And after all, seeing is believing. So we can merge up the phenotypic data with our molecular markers that are expressed on the surface. And so going back a step, these are some of the measurements that we take on each cell. So we look at the nuclear area, nuclear convex area, we look at the circularity, what the nucleus looks like, what does the DAPI staining look like, is there nuclear speckling, are there macronucleoli? So clearly we're looking at a lot of different features here, and this is going to paint a much more detailed picture of what the clonality looks like. So to demonstrate the utility of this, we wanted to look at each one of these features and see if we could come up with distinct phenotypic profiles for CTCs. And so we took 151 patient samples, which had about 9,400 some CTCs from patients at baseline, second line or third line therapy. And we can take these, all the measurements we made of the different cell phenotypes and characteristics, and we can do some principal component analysis to see which phenotypic features cluster together. And so the shown here is a 5x5 plot showing how these different features cluster and are interrelated. But the take-home is that this clustering can identify 15 unique CTC phenotypes. So now we have a specific phenotypic subtype of CTCs that we can go in and count. So back to that end, we have, we wanted to measure what are all the different types of phenotypic subtypes of CTCs that are present in patient samples. And so shown here, we were discussing those 15 phenotypic categories. So these are different cell types, and moving each column is a different patient. And where you see red, that's a high number of those cells, and where you see white, we didn't detect any of these cells. And so you can see that there's a lot of different phenotypic cell types present in each patient. And it's pretty hard to get a grasp on this, so one of the things we did is to look at, use a measurement of overall heterogeneity. And that's what's shown on top. We used a measurement called the Shannon Index, and this is something ecologists use when they go into rainforest to characterize the diversity of the different animals they see in the rainforest as a measurement of how ecologically healthy the environment is. And so you look at the Shannon Index of the different subtypes of CTCs we see by phenotype. This tells us how many different subtypes are present and what's the relative heterogeneity within a patient. And so if you look at this, they're ranked from low heterogeneity to higher heterogeneity at first line in blue, second line in green, and third line in red. We have some patients who are well beyond third line treatment, and we also segregated them into their own bucket. But what's interesting about this is that there's a lot of heterogeneity that exists regardless of the line of therapy. So this isn't just a result of therapeutic intervention. And so we can draw a cutoff and say that this is what we're defining as high heterogeneity, and that's the dotted line shown above. And we say, okay, if these patients are considered extremely high heterogeneity and the other patients are considered lower heterogeneity, how does that relate to their response to therapy? And so this is segregating those two patient populations, high heterogeneity shown in the red, and blue being the low heterogeneity population. And we can look at the relative time on drug. And so this means the time until they failed the drug, so when the therapy stopped working. And so you can see a statistically significant difference in those two patient populations in their response to therapy. And in general, the patients with lower heterogeneity were on drug for 5.7 months longer. And that's a pretty significant odds ratio of 11.6. So this is really highlights the fact that heterogeneity, and it also corroborates what we presented earlier, where they had shown that higher heterogeneity influences and negatively responds to targeted therapies. And so when you look at higher heterogeneity in response to AR targeted therapy, and again, these are showing the waterfall plots of PSA measurements after treatment. And so this is showing that their best PSA response, and when you see the lines go up, that means they didn't respond. PSA continued to increase and they were resisting therapy, and when you see the lines go down, the therapy had some effect and we reduced the PSA levels. And so when you look in the AR targeted therapy cohort shown up top, you can see our highly heterogeneous patients shown in red clearly cluster together in the resistant category. But when you look at response to taxanes, you can see that the highly heterogeneous patients both responded and resisted therapy. So this really speaks to the fact that understanding the subclonal heterogeneity prior to and throughout the treatment with targeted therapies is critical. So CTC heterogeneity, we wanted to further define these, so we've looked at protein and morphology, and we want to integrate all these together. We also can look at genomic heterogeneity. And this was a study, this is data from a study where, with one of our first collaborators, Genentech and ICR, where we wanted to look at what is the most reflective measure of the metastatic population. So if we take a bone biopsy from metastatic castrate-resistant patients and we characterize it for P10 status, what's more reflective of the current metastatic state of the patient sample? Is it the CTC blood sample? Can we get a better accurate description of metastasis in the blood sample? Or would it be better to go back to a primary archived tumor sample? And so we had each of these for each one of the patients, and they were all analyzed independently. And so we looked at P10 status, and in the CTCs, you can see that the P10 status, we found P10 loss, and we also found normal P10 cells within patients. Within the bone metastatic sample, we also saw similar heterozygous loss and normal wild-type P10 status as well. However, within the primary tumor sample, we saw predominantly the P10 normal. So this really says that P10 loss was a subclonal driver that happened later in tumor progression. And if we were to base our therapy on this, we would, and we looked at the bone biopsy, we would say that the patient's eligible for a PI3K inhibitor. If we were to say, look at the CTCs, we would also give him that same inhibitor. However, if we were to go back to a primary tumor sample, he would not be eligible for the PI3K inhibitor. So I think this really speaks to the fact that the CTCs are really reflective of subclonal heterogeneity and really reflect the active metastatic subclonal populations. And so when we look further into the CTCs that we see in these patients, what you can see here is each column represents the percent of CTCs with a particular P10 status. So P10 loss is shown in maroon, P10 heterozygous P10 loss is shown in gold, and P10 loss or P10 wild type is shown in gray. And so when we look across the patient cohort, you can see that even when we see P10 loss, that's still a subclonal population. When we see P10 heterozygous loss, that's still a subclonal population. So it really speaks to the power of looking at single cells and understanding the relative proportions of cells that are actively metastatic with a particular tumor suppressor loss. If we look at how well the CTCs would match up and predict the P10 status of the fresh bone biopsy, the positive predictive value was 79%. Whereas if we look at how well the archival tumor sample would predict the P10 status of the bone biopsy, then we would see that the positive predictive value drops considerably to 53%. So this is, again, confirming that the CTCs are reflective of the active metastatic population. So something we're working on now, and we developed and launched in the earlier part of the year, was a NGS-based assay to globally profile genomic alterations across the whole genome. So this is we isolate single CTCs. We can then do low-pass whole genome sequencing, and then we can look at tumor suppressor or oncogene copy number alterations across the entire genome. So we can then take those copy number profiles and we can cluster them together in very distinct clonal populations. And then because we look at cell morphology as well as protein and molecular markers, we can collate all that data together to come up with very distinct subclonal populations and then track those throughout the course of treatment. So to look at this clinically, we looked at 350 CTCs across 17 separate patient samples. And these are patients who, again, were either resistant to AR-targeted therapy or responsive, or resistant to taxane therapy or responsive. So this is an example of some of the typical data that we see. And shown on the right are copy number profiles of each CTC. And what's really cool here is you can see that the copy number profile is paired up with the actual image of the cell. So you can look, it's kind of hard to see on these slides, but you can actually correlate what the cells look like and to what their genomic profiles look like. So we're actually starting to be able to look at phenotype to genotype associations. And so what jumps out at you when you look at these two distinct patient examples is on the left is a responder. This guy is responding to AR-targeted therapies. There's very little clonal diversity. He has, mostly a single or maybe potentially two different clonal populations. However, they're relatively flat in copy number alteration. When you look at the patient on the right, we see that you can detect a larger number of distinct clonal populations by clustering. And wherever you see red are areas where we see deletions, and wherever you see green are areas where we see amplifications. So when you look at each CTC, you can see there's some common drivers associated with this, the clustering of these cells together in different populations. And of particular interest, if you look over to the right, on the very right-hand side, you can see each one of these cells, almost all of the cells in this patient had an amplification of the AR gene. And so this is an early alteration that was very early. And so if we looked at this in a heterogeneous population, we would say it must be AR amplification that's driving disease. However, when we look at the different subclonal populations, we can find different subpopulations that have different tumor suppressor loss or oncogene gain. And to show you that across the entire 17 patients tested, we've broken up the patients by either resistance on top in the blue, where we look at either AR-targeted therapy resistance or taxane resistance, or on the bottom, patients responding to AR-targeted therapy or responding to taxanes. And we've looked at how many, across all the CTCs, which percent of the CTCs had subclonal populations of these different tumor suppressors, loss or oncogene amplifications that have often been associated with prostate cancer disease progression. And so interestingly enough, you can see that the majority of subclonal heterogeneity is in the resistant patients. And if we look at particular alterations, we can see that not only does a patient have AR amplification, he also has loss of RB, BRCA2, and ATM loss. So there's a lot of interesting subclonal late alterations that are happening that could potentially be driving that clonal differentiation. If we only looked in heterogeneous or populations where we would just pool all these cells, we would lose a lot of the resolution to see this. So I think at this point we wanted to wrap it up, and I'll just kind of go through and summarize a lot of the points that were previously made. So intra-tumor heterogeneity is frequently observed across multiple tumor types. I think that was the Swanton paper clearly says that, as well as other types of other sources. We have increased heterogeneity, definitely has a negative effect on targeted therapies and therapeutic response, and so this is been cited by others as well as observed in our own data. The CTCs are truly reflective of the subclonal populations and the active metastatic subclonal populations. And the EPIC platform's unbiased approach allows us to see a lot more, a wider biological range of CTCs, and integrating these molecular, phenotypic, and genotypic assays gives us the capability to really identify distinct subclonal populations that we can then target actionable markers to better stratify patients in metastatic disease. And so that wraps it up. And so I think at this point, we will open it up to questions.
Thank you for that informative presentation. Before we get started on the question and answer session, we'd like to ask you a couple of poll questions. The first one is, I am currently evaluating technologies that will enable me to utilize a liquid biopsy in cancer evaluation. Yes or no? you a minute to answer that one. Okay, and I'll just get a little ahead here and mention the next one. The next one is, would you like to be contacted by one of our technical team to discuss your project? Yes or no? Okay. I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, how does this technology of detecting and analyzing CTCs compare to cfDNA?
That's a great question. Both technologies, both cell-free and looking at CTCs, have strengths and can have some clinical utility. However, that being said, the DNA that you're looking at in cell-free DNA is largely the cells that are dying. And so when, there's some distinct differences that I think are important. When you're treating a patient, you want to see the emergence of resistant clones. Those are not the cells that are actively being targeted and that are dying. Also, there's a distinct advantage to single-cell approaches where we actually retain information of the cell of origin. So are we looking at a single cell with multiple mutations in a single pathway, or are we looking at multiple subclonal populations with individual mutations in different pathways? This is going to change how we prescribe targeted therapies, because if we're targeting particular pathways, we need to know what the clonal population is that is defective in that pathway. And then finally, I think that one of the big differences is thinking back to that example of the individual fruits all in a mix versus the kind of fruit smoothie that we saw, where you can see that when you homogenize all these cells together, you lose sensitivity to see copy number alterations, because now you're going to see a lot of cells, like in our case of our P10 study, there's a heterogeneous population of cells that have P10 loss, whereas there's a lot of cells that didn't have the P10 loss. So I think you would lose the resolution to see that, and that's potentially a relevant therapeutic target.
Great. Can EPIC perform cfDNA tests as well as your CTC analysis on the same sample?
Yeah, absolutely. I think at EPIC we really own the sample. When we get our sample in, we're currently in the taking, in a lot of studies, we take a sample off the top to isolate cell-free DNA from before we process them with our standard assay. And this gives us the flexibility to pool it and do comparative analysis, but also to look for actionable markers within the cell-free DNA that might direct patient treatment.
What other genomic assays can EPIC perform besides CNV?
So we're currently in the process of developing hotspot mutational NGS assays, and we also have the capability to run qPCR for specific point mutations. There's a lot of great qPCR tests that measure a single nucleotide variant, like KRAS or EGFR, but we also have the ability to look at thousands of these mutations together in a single targeted resequencing pool, and we still retain the single cell resolution, so we can look at how many cells within a particular patient are harboring, a T790M resistance mutation throughout the course of treatment with a TKI.
Can you provide a list of assays and biomarkers you can test for?
Absolutely. So I think we're happy to follow up with anyone and provide a full list. I think the information shown here on the screen, you can just email for information at EPIC, and we'll follow up with you to give you a more detailed list of the different assays we can offer and how we can work with you to better characterize your samples.
When will the IVD instrument you mentioned be available?
So I think we're targeting next year. I can have, there's probably a more detailed plan that I'd be happy to have some of our business development associates follow up with you, but we are currently in the process of IVD development, and that is ongoing, and we're hoping to have that pieces of that platform really soon.
Have you compared the number of CTCs EPIC identifies with other CTC enrichment platforms?
Yeah, that's a good question, and one we get all the time. So we currently, the FDA cleared assay, the Veridex cell search assay, we're used for CTC enumeration. We constantly do comparisons versus that assay. Although we're really doing a different process where we're actually characterizing the cells in much more detail rather than just enumerating them. But when we do just the comparisons of enumeration in a side-by-side setting, we consistently detect more CTCs, and this goes back to the enrichment-free analysis. So we're not discarding any CTCs based on physical properties such as their epithelial marker expression.
And this next one is a two-parter. Have you seen a correlation between CTC heterogeneity and tumor progression, or does the CTC population tend to be more or less heterogeneous in patients with aggressive cancers?
Yeah, that's a good question, and I think there is a correlation between CTC heterogeneity and tumor progression versus the targeted therapies. And I think we showed some data showing that when there was higher heterogeneity of different subclonal populations, they responded more poorly to therapy. And we also have, there is also, we'd be happy also to follow up with you offline if that's, to give you more information about those associations.
How did you pick your single CTC for sequencing, and are they fixed cells on a slide?
Yeah, so that's a great question. So we do this single cell isolation following our standard EPIC process. So the blood comes in, it gets deposited on cells or on slides. Then we record while we're scanned the XY coordinates of every single cell. So then we can go back to the slide after we've scanned and characterized all the morphological and molecular markers on those cells and recover those CTCs. We have a unique process for doing that. But it drives basically, it's an automated or semi-automated process where we go and we can recover the CTC. Then it is dropped into a single well of a PCR plate, and then we have a single cell isolated in a single well, and we can proceed with genomic analysis.
Looks like we have time for one more question. Have you ever used this platform to look at cells in healthy normal samples?
Yeah, so we routinely run that as a control. That's a great question. We routinely run healthy donor samples as a control, and this is we don't see circulating tumor cells in these samples. So, whenever we get healthy donor blood, we do a lot of spike recovery samples, and we do not see a lot of circulating or any circulating tumor cells in most of these samples.
Okay, turns out we have time for a couple more. So what is the fraction of single-cell CTCs that yield evaluable NGS data, low-pass CNV?
Oh, that's a good question. So recently in a study, I think our success rate is upwards of 90% from the cells we targeted to pick versus the cells that we get QC profiles that pass our library complexity as well as our sequencing QCs. So we're about 90%, 95% success rate.
Okay. Can you do RNA assays as well as the DNA ones?
Yeah, that's a great question. So currently, we're developing our several RNA assays. So we have RNA fish that we're currently developing targeting several transcripts, and we have a few assays that are starting to get close to through the development cycle. We're also in the process of developing small targeted digital gene expression, using qPCR. So we're looking at can we lyse open a single cell and look for the relative expression of 6, 7, 8 genes from a single cell. And so the early data in development, we're having some success with it, and we think the early data looks positive, but we're still in development with that capability.
Okay, here's your last question today. What is the depth of sequencing needed for CTCs or primary metastatic samples for clonality and subclonality?
So to detect in the primary tumor sample, most of the reports that were shown here, the sequencing depths were upwards of 500x to detect the subclonal population. So they really sequenced very deeply, especially in the patient, the colorectal example where they sequenced the primary tumor. They actually had to go back to it and sequence deeper to get the detection to be able to detect those subclonal variants. With the CTCs, we don't have to sequence as deeply because we're doing the analysis from a single cell. However, we do have to make sure that we get uniform coverage over the areas we're trying to profile. So currently in our C&V assay, we get about 0.1x coverage across the genome, so it's low pass whole genome. For targeted resequencing applications, we can shoot for a 50x coverage just to even out all the different, the uniformity of coverage across the different regions of interest.
Well, thank you. Sorry, we have run out of time. We'll follow up with you on questions we did not get to today in the next couple of days. I'd like to thank our sponsor, Epic Sciences, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through December of this year. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.